So it's my pleasure to introduce Luca. Luca is a CCAS DARE fellow, and prior to becoming a postdoc in my group, he was a graduate student with Neil Garg at UCLA, um, and he's going to be talking about a project that was actually initiated by Logan in our laboratory, along with Lucas from the Sigman laboratory, where we're looking to identify what might be important skeletal editing reactions that have yet to be realized. And so, Luca, the floor is yours.
All right. Well, thanks so much, Richmond, for the intro there. So since I'm a little bit new in the Sarpong group and also in CCAS, I'll be talking about, yes, as Richmond mentioned, some chemistry that was developed earlier and published earlier this year. And so this was chemistry developed by Logan here from Richmond's group, as well as Lucas in the Sigmund lab, and also in collaboration with chemists at Merck. And so this was really focused on this emerging area of skeletal editing that has seen a lot of growth in the last 5 years. And the concept here is that you can take core structures of molecules and go over to and hop over to different core structures. So for example, with this heterocycle here, you could do an atom swap, switching the heteroatom for a carbon to access benzene rings. You can also do insertions and deletions to expand or contract rings, as shown here. And you can even do transmutation reactions where you replace heteroatoms with other heteroatoms. And this allows you to do late-stage modifications of heterocyclic compounds. And it turns out that if you look in the literature for all of the reactions that I just described, there are examples in the literature. And so as the field continues to grow and to progress, one question is how do we decide which new reactions we want to invent and which ones are going to be the most impactful? And of course, impact, we can define this in many different ways. But for this study, impact was really just defined as what reaction would allow you to explore the most new chemical space. And so for this, the team developed a Cheminformatics workflow. And this starts off by looking into libraries of compounds. And they looked at a couple of different libraries, but the one I'll focus on is the enamine library, which had the most compounds. And it starts off by sorting these structures and looking at which heterocycles are present. So for example, this pyridine core was found in over 18 million of the structures in this data set. And I think this really highlights the value of using computer tools here because, for a human to go through 18 million different structures, it'd be quite difficult, but for a computer, it's a little more straightforward. And so from there, in the second step, this was kind of the key point of this study, was that they did virtual core atom transformations. So what this means is you would take a structure in the data set, and then keeping the backbone and the substituents the same, the core was modified to access a different heterocycle. So for example, you could do a carbon to nitrogen swap to the pyridazine here, or a carbon deletion to get to these perols. And importantly here, again, the substituents stay the same, but the core here changes. And then in the last step, the new structures were compared and it was assessed whether the new compounds made were already known structures in the chemical literature or not. And so if we look at this example here, for the pyridazine, it turns out that 75% of those new pyridazines were already known structures in the chemical literature. On the other hand, if you look at the new perols here that were made in silico, and now only 26% of those new structures are known. And so the conclusion you could arrive to here is that the pyridine to perol skeletal edit would allow you to have a greater exploration of chemical space because now more of those new structures that you're making are actually new compounds. And so this was done for all of the heterocycles shown here. And at the end, kind of in the conclusion to the paper, they laid out the top 5 single atom skeletal edits from this enamine data set. And so, for example, if you were to be able to develop pyrimidine to imidazole ring contraction via carbon deletion, this would really allow you to explore a lot of new chemical space as only less than 12% of the new structures here have previously been characterized. And so the Sarpong group has been interested in developing these kinds of skeletal edits, and I won't spend too much time on this, but I'll just highlight a couple of cool examples. The first is this nitrogen atom phototransposition, which allows you to take these indizoles and upon irradiation, transform them into these benzimidazoles. And additionally, these carbon atom deletions allows for the conversion of pyrimidines to pyrazoles through treatment with trifluc anhydride followed by hydrazine there. So to summarize here, I talked about kind of this area of skeletal editing and how we can start thinking about new kinds of reactions that we might want to develop. And a cool study that took a cheminformatics approach to kind of sort out these structures, do these virtual core atom transformations, and then look at how many of the new products made would be actually new compounds. And so I think it's a cool way to kind of use cheminformatics to think about how we want to design and develop new chemical reactions. So with that, I'll thank the Sarpong Lab for the really warm welcome they've given me in the last month or so. Of course, CCAS as well for welcoming me in, and Merck who collaborated on this paper, and I'd be happy to take any questions.
All right. Thank you, Luca. There's already one hand up. Paola.
Yes, I'm wondering here, not all of these edits are equally successful. They are obviously differently robust. Can you make any kind of estimate for the known edits? How frequently will they fail and how frequently will they succeed on the diverse cores you have listed here?
Yeah, thanks for the question. I think this will definitely be dependent on the reaction and the core structure. So I'm not sure if I can make an exact prediction. And of course, for new reactions that are being developed, it's hard to predict. Richmond, if you want to comment, if you have any thoughts as well.
Yeah, I can certainly comment. I mean, this is going to be a heavy lift from a cheminformatics standpoint. But certainly for each of the reactions that we develop, we do engage our partners in terms of thinking about either classifications or optimizations. But these are all dependent, on reagents and conditions for each transformation. We haven't looked at it wholesale for, the 18 million transformations, for example, that were listed here. But we do look at it for each individual transformation. And so we published, for example, on an isotopolog synthesis of going from N14 to N15. We looked at a classification model with the Sigman laboratory in order to predict efficiencies for those reactions. But each individual transformation in itself is quite a heavy lift. So, but it's a great challenge that you're posing to us, Perla. So maybe something that we can look at as a center. So appreciate the question.